Chapter 3, the cellular level of organization. So as we get into this chapter, we're going to start with all the parts of the cell. We can divide the cell into three basic parts. Number 1, the cytoplasm. This is a phospholipid bilayer that's going to be arranged like almost a fatty sandwich. We'll look at this and explain this analogy in a little bit. The second part is going to be the cytoplasm. It will contain the cytosol and all the organelles sustained and supported in there. And the third part is the nucleus, which will contain the chromatin and the nucleolus. This is the brain of the cell, all the genetic information. Let's take a look at a picture of a cell so we can identify and discuss all the parts. So we'll start in this upper right corner and look at the nucleus. Again, it contains the chromatin, nuclear pore, nuclear envelope, nucleolus, and this will be considered the brain of the cell. So it tells the cell what it's scheduled to do. Our second part here will be, we'll put a 1 by the nucleus, our second part will be the cytoplasm. This is going to include the cytosol plus all the organelles except the nucleus, and we'll talk about those organelles in a little bit. Now as we move our way away from the nucleus, we're going to start outlining some of these organelles, which is Latin for little organs, and explain what they do. Our first is going to be our rough endoplasmic reticulum. Now if there's an RER or rough endoplasmic reticulum, there will be a smooth. So if we move over to the left, we see over here a smooth endoplasmic reticulum. Now what do these guys do? The function of the rough endoplasmic reticulum is to produce proteins. So all those ribosomes that are studded to the surface of this membrane is producing proteins. The next structure we're going to see is the Golgi apparatus. Now the function of the Golgi apparatus is to package all those proteins and sort them and put them into nice, neat little packages to be shipped out of the cell. And we can see those little vesicles being created and then moved and then exocytized out of the cell. A couple other things we're going to see is going to be some microtubules, mitochondria, right, the powerhouse of the cell. So we've talked about mitochondria being the powerhouse of the cell. We come back to our smooth endoplasmic reticulum. This is going to be for fat production. We're going to make fat here that's going to help replenish that phospholipid bilayer. Our seventh organelle is going to be a lysosome. These are vesicles that contain digestive enzymes. So when a cell has to dissolve or destroy something or digest something, especially if the cell is phagocytizing it into its body, a lysozyme will attach to that residual body and digest its contents. Next is our plasma membrane, phospholipid bilayer. We're going to spend some considerable time talking about this in the next few slides. From there, we're going to hit up our centrosomes, right, which are going to have our pericentriolar material and our centrioles. These are the spiders of the cell. They spin a web, especially during mitosis, to attach to the center of the kinetochore of our genetic material to rip it apart for cell division. We have some intermediate filaments. We have some microfilaments, which are going to extend through some of our cellular projections. Some microtubules, which also give rise to our last cytoskeleton. This is what gives the cell its shape. It's going to allow for cells to have, again, various shapes. They could be flattened, they could be boxed, they could be columnar, they could be round. So the cytoskeleton, like the skeleton of you, gives you shape, will give the cell a shape. A couple more projections of the cell membrane. We deal with flagella and cilia, or cilium. These guys will give hair-like, cilia will be the hair-like projections on the cell membrane, where flagella, we see the only flagellated cell structure in the human anatomy is sperm, will give for motility. Our cytoplasm. Cytosol is also known as the intercellular fluid portion of the cytoplasm, so it's what's inside the cell. Organelles are specialized little structures that have specific shapes and perform specific functions, what we just outlined in our previous slide. That brings us to our plasma membrane. The plasma membrane is a flexible yet sturdy barrier that surrounds and contains the cytoplasm of the cell. It is a phospholipid bilayer. So we have these heads and tails. that are arranged in an opposite conformation. So we have our polar heads and nonpolar tails. These are our hydrophilic and hydrophobic regions. So these are philic, loving, water loving, so hydrophilic regions, whereas those nonpolar tails are going to be hydrophobic. So as you can see our analogy before where we said it is a fatty sandwich, our polar heads are going to be the bread, and this fat-filled center will be the middle part of our sandwich. Now, a couple things to notice here. We are setting up a barrier, a boundary to the cell, and we see a couple things inserted into it, like proteins. We see a glycocalyx. We see channel proteins with pores. We set up the boundary from the inside of the cell to the extracellular fluid on the outside. We also see some integral or transmembrane proteins that don't set up a pore but will activate other second messenger systems. So we will talk about all these different structures and exactly how they contribute to the fluidity or structure of the cell. The last thing to point out is right here, cholesterol. And we'll talk about cholesterol in a little bit, but notice that it is integrated within that fatty sandwich and it plays a real pivotal important role. Let's look at the phospholipid portion of our cell membrane. We already said that the polar head is hydrophilic and that our nonpolar tail, so here hydrophilic, and we said our nonpolar tails were hydrophobic. Now, we're not going to go too much into detail with how these are arranged with all the carbon chains, but phospholipids are an important component of the cell membrane. They are setting up the cell to establish concentration gradients from the intracellular fluid to the extracellular fluid, allowing stuff to move through. Remember, one of our components of the plasma membrane is that it is semi-permeable. So this is an important characteristic of our cell membrane. Let's now look at those membrane proteins. We have two types of proteins. One is an integral protein. They're also called transmembrane. The job of these integral proteins is to be inserted between the outside and inside of the cell, allowing for communication of either hormones or chemicals or setting up electrochemical gradients from the outside to the inside of these cell structures. Next are peripheral proteins. Again, these proteins are going to lie on the periphery or on the outside or margin of the cell, and we'll talk about what these guys do in a little bit. Functions of membrane proteins. Membrane proteins can serve a variety of functions. The different proteins help determine many of the functions of the cell membrane. So let's take a look at them. Ion channel, which are integral proteins. They form a pore through which a specific ion can flow to get across membranes. Most plasma membranes include specific channels for several common ions. These ions include sodium, potassium, and chloride. So if you think about the charge on a sodium potassium, they're positive, on chloride would be negative. So what are we doing here? We're setting up potentials, electrochemical gradients, and we can manipulate these gradients, manipulate the charge in the cell and outside of the cell to favor propagation of things like action potentials, especially on a nerve cell. The next is a carrier membrane protein. This is another integral protein. It functions to transfer a specific substance across the membrane by undergoing a change in shape. For example, amino acids, these are needed to synthesize new proteins, so they enter the body cells via carriers. Glucose would be another one, and sodium-potassium pumps would be another one. Carrier proteins are also known as transporters. The next is a receptor. They're also called ligand-gated channels. These are another integral protein. They recognize specific ligands and alter cell function in some way. For example, antidiuretic hormone binds to receptors in the kidney and changes the water permeability of certain plasma membranes. Also, for another example, on a cell body, we can bind a specific ligand, meaning acetylcholine, which would help depolarize the cell, specifically at the cell body of a neuron, to allow for graded potentials to summate to propagate action potentials. When we do neuro, we'll cover this in much more depth. Next are enzyme membrane proteins. They can be integral or peripheral. They catalyze reactions inside or outside of the cell, depending on which direction the active site faces. For example, lactase protruding from epithelial cells, from cell lining your small intestines, split the disaccharide lactose in the milk you drink. So we have enzymes that are breaking many sugars into a simple sugar so that you can digest it more easier. Linker proteins are integral and peripheral. These are anchors, filaments inside and outside the plasma membrane, providing structural stability and shape for the cell. They also participate in movement of the cell or link two cells together. And we're going to name some of these linker proteins when we talk about desmosomes and hemidesmosomes and see exactly how they do that. The next are MHC proteins. These are identity markers. These are how cells identify themselves. They are glycoproteins. It's the way your body distinguishes your cells from anyone else's. So it's a major part of our histocompatibility or immune system. Membrane fluidity. Our membranes are fluid structures because most of the membrane lipids and many of the membrane proteins move easy in the bilayer. Membrane lipids and proteins are mobile in their own half of the bilayer. Cholesterol, as we pointed out in the previous slide, serves a super important function. Its function is to stabilize the membrane and reduce the membrane fluidity. So another picture to look at some of these structures. Again, we see cholesterol here. We see our sodium channel here, our ball and chain. So these are sodium channels. We have some potassium channels and some more transmembrane second messenger system. So these activate second messenger systems. And again, don't worry too much about the terminology here. We're gonna get into detail with what these systems are later on throughout this semester. Now we said that the plasma membrane is deemed or termed semi-permeable. So let's talk about that permeability. Plasma membranes are selectively permeable. The lipid bilayer is always permeable to small, nonpolar, uncharged molecules. Transmembrane proteins that act as channels or transporters increase the permeability of the membrane. So when we said here before, uncharged molecules, we're not talking about sodium, potassium, and chloride. Right? These are charged molecules. So that's what we need those transmembrane proteins for, to help move those charged molecules through. Macromolecules are only able to pass through the plasma membrane by vesicular transport. So anything large has to be phagocytized in or endocytized in and then move through a vesicle. Now we hinted to gradients across the plasma membrane. We've hinted to electrical gradients and we've talked about briefly concentration gradients, so let's put these together. So we're talking about making up these electrochemical gradients. So how do we do that? So a concentration gradient is the difference in the concentration of a chemical between one side of the plasma membrane and the other. So we're talking about internal environment inside the cell versus external environment outside the cell. So intracellular versus extracellular. An electrical gradient is the difference of concentration of ions between one side of the cell, the plasma membrane on the outside, and the inside. So a lot of these gradients are maintained by those channels that we just got finished talking about. So a cell, when at rest, is termed to be at resting membrane potential, where we're maintaining a consistent electrical gradient of about minus 70 millivolts. So inside the cell, if we put a voltmeter inside that cell, would measure about minus 70 millivolts. So we're setting up those electrical gradients. And then the concentration gradients, again, we could vary based on how we move things in or out of the cell. Transport across the plasma membrane. Transport processes that move those substances that we just got finished talking to across the cell membrane go via two ways: passive processes and active processes. Passive processes include simple diffusion, facilitated diffusion, and osmosis. Table 3.1 that we're going to look at in a couple slides is going to explain in detail some of these processes. Active processes. So if we think about active, we're relying on energy. So we need energy to get this movement to occur. So we deal with active transport and vesicular transport, whereas passive processes require no energy. On your PowerPoint slide, you can click on this link to bring you into Wiley to show you a little animation about the transport across plasma membranes. Table 3.1 as promised. So let's look at some of our passive processes. So the description, we're moving substances down a concentration gradient until equilibrium is reached. This does not require cellular energy in the form of ATP as opposed to active processes. We start with diffusion, right? So we're moving molecules or ions down a concentration gradient due to their kinetic energy until they reach equilibrium. That's the key here. We want to reach equilibrium. The first is simple diffusion. Passive movement, so again, no energy, of a substance down its concentration gradient through the lipid bilayer of the plasma membrane without the help of membrane transport proteins. So, what are some examples of things we're moving? They're nonpolar hydrophobic solutes like oxygen, carbon dioxide, fatty acids, fat-soluble vitamins, nitrogen gases, water, urea, and small alcohols. For facilitated diffusion, this is passive movement of substances down its concentration gradient through the lipid bilayer with the help of transmembrane proteins that function as channels or carriers. So these are those charged ions that we were talking about. So polar or charged solutes like glucose, fructose, some vitamins, and our main ions, potassium, chloride, sodium, and calcium. Osmosis is a passive movement of water molecules across a selectively permeable membrane from an area of high to low water concentration until equilibrium is reached. So again, osmosis is pretty straightforward. We're dealing with a solvent. What solvent? Water. And we're moving it from high to low concentration until we reach equilibrium. Let's put this to picture. Let's talk about simple diffusion and what can influence the rate at which diffusion occurs and how we reach equilibrium. So we start with A, the beginning. So we see all the blue dye at the bottom. Over time, it's going to start diffusing upwards to reach equilibrium until we hit full equilibrium where that blue dye is equally distributed through the entire cylinder of water. Now, what can affect this? What can change the rate at which diffusion can occur, the rate at which equilibrium can be reached? One, the steepness of the concentration gradient. So how much blue dye do you put in there? How many molecules have to diffuse across this distance? Temperature. The colder the solvent, the longer it's going to take, because molecules are moving slower, to move and reach equilibrium. The warmer, the faster. Mass of diffusion substance. The heavier those substances are, they're going to tend to settle to the bottom of that tube and will resist equilibrium. Surface area. So the total distance in this case that solute has to move through that solvent, the distance determines how long it takes to reach equilibrium. And then again, the diffusion distance works with that surface area with regards to how much time it takes to reach equilibrium. Facilitated diffusion. Again, those transmembrane proteins help solutes that are too polar or too highly charged move through the lipid bilayer. Again, that sodium, that calcium, the potassium, the chloride, right? The processes involved are channel-mediated facilitated diffusion or carrier-mediated facilitated diffusion. We can look at channel-mediated facilitated diffusion. This channel is called a ball and chain. So what's happening? The gate is open and potassium is able to move out. When the gate is closed, potassium is not allowed to move out of the cell into the extracellular fluid. So what determines if this gate is open and closed? It's charge. It's all about charge. So by depolarizing a cell, making it more positive, will allow this gate to open and move potassium out. Carrier-mediated facilitated diffusion. As we said before, here's our example of glucose. When glucose binds to that substrate-specific channel, the channel will open and drop glucose off into the cell. So again, we need a concentration of glucose in the extracellular fluid. It binds like a lock and key into the cell into that transmembrane protein, which will allow it to have this conformational change, bringing more glucose into the cell until we reach equilibrium. So let's put it all together. So let's look at a comparison of diffusion. So we're dealing with concentration gradients. So we see outside the cell we have a high concentration compared to the low concentration inside the cell. So how do we get it in there? What are our three processes? Again, one, simple diffusion. And you can see that little guy is just moving right through based on concentration gradient from high to low. Now we deal with channel mediated. So ions are going to move into, again, from a high to low concentration, but with help of a depolarizing event spreading across the cell membrane. And carrier-mediated facilitated diffusion, like glucose, working its way in like a lock and key, opening that membrane, that transmembrane protein, and dropping glucose into the cell. Next is osmosis. Again, remember moving water to high concentration to low concentration, but now let's throw in a solute. So when we talk about osmosis, it's the net movement of water through a selectively permeable membrane, i.e. our cell membrane, from an area of high water but low solute concentration to an area of low water but high solute concentration. So let's have an example. We have a tube here, a left arm and a right arm. We have our selectively permeable membrane right here. We have an area of high concentration on the right side and a low solute concentration on the left side. So the question is, what's going to happen? Which way is water going to move? So look at the wording above. It is a selectively permeable membrane from an area of high water but low solute to an area of low water but high solute concentration. So here's our key. We're moving to a high solute. So water is going to go to the right. And you can see that. Look in the right arm. The water climbed up that cylinder, and this is due to hydrostatic pressure. Now, where can this and how can this reach equilibrium? So we can restore equilibrium by applying a pressure. This is going to be equivalent to our osmotic pressure in our body. So we apply a pressure that's going to help push water back through that semi-permeable membrane, again, regardless of solute concentration, all these little triangles here, and reaching equilibrium. Tonicity. Tonicity of a solution relates to how the solution influences the shape of body cells. So we're talking water, we're talking solutes. So let's look at a red blood cell. If we look at A, the red blood cell, a typical biconcave disc, water is moving freely in and out of the cell, and we're maintaining a normal shape. Isotonic means equal or same, right? So with an equal distribution of solutes and water, the cell remains relatively untouched as far as shape is concerned, and water is freely moving. We're at equilibrium. Now, what happens if we take that same red blood cell and throw it into a hypo? Remember, hypo means low. A hypotonic environment. So the red blood cell is going to undergo this process called hemolysis, so cell destruction. So let's think about what's happening. So inside the cell, we have a high solute concentration inside the cell. And as we said before, we're going from a low solute to high solute. So water is moving into the cell. So what's going to happen? All that water is rushing into the cell is going to rupture and die. Now, let's flip it. Let's look at a condition where we have a hypertonic solution. So hyper means high. So what's happening here? So the solute concentration inside the cell is low. And that's problematic because water is going to try to equilibrate by shoving it out of the cell into the beaker, again trying to reach equilibrium. So the cell is going to shrivel. That shriveling is known as crenation. Our next table takes us through all of active processes. So again, remember with active processes, we are moving substances against a concentration gradient, which is going to require cellular energy in the form of ATP. We have active transport. This is an active process in which a cell burns or expends energy to move a substance across the membrane against its concentration gradient by transmembrane proteins. That's going to function as those carriers. We're dealing with polar or charged solutes. We have two types of active transport. We have primary active transport and secondary active transport. Primary active transport are active processes in which a substance moves across the membrane, again against its concentration gradient, by pumps or carriers that use energy supplied by hydrolysis of ATP. So we're going from ATP to ADP. That release of a phosphate is that one energy that we need to power this pump. The ions that we're moving are sodium, potassium, calcium, hydrogen, iodide, and chloride, and a few other ions. Secondary active transport are coupled active transport of two substances across the membrane using energy supplied by a sodium or hydrogen concentration gradient, maintained by primary active transport. So we deal with antiporters and symporters. So antiporters move sodium or hydrogen and other substances in opposite directions, hence anti, against, across the membrane. Symporters move sodium or hydrogen and other substances in the same direction. So sim means same and anti means opposite or against, so in the opposite direction. So glucose amino acids are going to be moved into the cell in conjunction with calcium, hydrogen moving out of the cell with their sodium or hydrogen counterparts. Next is transport in a vesicle. So we have active processes in which substances move into or out of a cell and vesicles that bud from plasma membranes. This requires energy as well, supplied by ATP. Here are our processes for vesicular transport. Endocytosis. Endo means within, cyto, cell. So we're moving substances into a cell via vesicles. We can go by receptor-mediated endocytosis, where there's a ligand receptor complex that triggers enfolding of a clathrin-coated pit that forms a vesicle containing the ligand. So what ligands are we talking about? Transferrin, low-density lipoproteins, LDL, a form of cholesterol, some vitamins, certain hormones, and antibodies. Phagocytosis. Phagocytosis, cell eating. This is movement of a solid particle into a cell after pseudopods, these fake arms, reach out, engulf that foreign substance like a bacteria or virus, and forms this phagosome within the cell. At which point that lysosome that we talked about before will attach, forming a phagolysosome to digest that foreign object, that bacteria, that virus, aged or dead cell. Bulk phase endocytosis is cell drinking, also known as pinocytosis. This is movement of extracellular fluid into a cell by infolding of plasma membrane to form a vesicle, hence cell drinking. What are we moving? Solutes in the extracellular fluid. Exocytosis. So if we are endocytizing, bringing stuff in, we have to kick it out. So movement of substances out of a cell in secretory vesicles that fuse with the plasma membrane and release their contents into the ECF, extracellular fluid. Things like neurotransmitters, hormones, and digestive enzymes. Transcytosis. So this is movement of substance through a cell as a result of endocytosis on one side and exocytosis on the opposite side. So trans means across. We're moving across the cyto cell, so across the cell, utilizing both endocytosis and exocytosis. So what are we doing this to? Substances such as antibodies, moving across endothelial cells. It's a common route for substances to pass between blood plasma and the interstitial fluid. Let's put it to picture. Primary active transport. Energy derived from ATP changes the shape of a transporter protein, which pumps the substance across the plasma membrane against its concentration gradient. So, we look at our sodium-potassium pumps. It works on a 3:2 ratio. 3 sodiums out, 2 potassiums in. So 3 sodium ions from the cytosol bind to the inside surface of the sodium-potassium pump. Sodium binding to that pump triggers ATP to bind to the pump and then to be split into ATP and phosphate. This is our hydrolysis. The energy from that split, from ATP splitting, causes the protein to change shape, which then moves the sodium out of the cell. Two potassium ions land to the outside surface of the pump and cause that phosphorus to be released, the one that we just got from the hydrolysis of ATP. The release of that phosphorus causes the pump to return to original shape, which then moves the potassium into the cell, a 3:2 ratio. Secondary active transport. Energy stored in a hydrogen ion or sodium ion concentration gradient is going to be used to drive other substances against their own concentration gradient. So we see that we have high sodium on the outside, lower sodium on the inside, driven by things like these calcium gated channels. We have glucose gated channels and amino acid gated channels. Again, we have a lock and key mechanism here with these glucose and amino acid. Once that substrate binds specifically to this channel, it will signal the opening. Active transport in vesicles, receptor-mediated endocytosis. So here we go with a LDL receptor-bound complex. LDL arrives, it binds to that receptor. The cell invaginates in, the plasma membrane invaginates in, invaginating clathrin-coated plasma membrane, and forms this vesicle. It is a clathrin-coated vesicle. Now, the clathrin is removed, at which point this uncoated vesicle will fuse with an endosome. That endosome will degrade, right? So the endosome will form this vesicle that will transport and combine with the lysosome that will be broken up and degraded and then kicked back out into the cell. Now, we can also recycle we can recycle these receptors to the plasma membrane. So those receptors are recycled and then kicked back onto the surface of the cell membrane. So our cells truly are master recyclers. Next is phagocytosis. So what happens? We find this little microbacteria that will be gobbled up with these pseudopods, these little fake arms that are going to reach out and form a phagosome. Once that phagosome is within that cell, a lysosome will come attached, forming a phagolysosome. So a fusion of the lysosome and the phagosome. Once in there, those digestive enzymes will digest that microbe or that bacteria and form a residual body, at which point this will most likely be kicked out through exocytosis. Cell drinking or pinocytosis, also known as bulk phase endocytosis. So, solutes are drunk into the cell. We form a vesicle. Fluid and dissolved solutes are located inside that vesicle. The lysosome again comes in, forming this fused structure, at which point those lysozymes will become active and digest those solutes, dissolved solutes inside that vesicle, and kick it out into the cell as digested solutes. Active transport in vesicles, exocytosis and transcytosis. Exocytosis, again, membrane-enclosed secretory vesicles fuse with the plasma membrane and release or kick out their contents into the extracellular fluid. Transcytosis is a combination of that endocytosis we just got finished talking about and this process of exocytosis. So it's used to move substances through the cell membrane on one side of the cell, across it, and out of the cell membrane to the other side, effectively causing for the digestion or the removal of viruses, bacteria, pathogens, or other like substances. Table 3.1 is a review of what we talked about on our first few slides. This is the active processes for Table 3.1, summating what we've talked about in this entire lecture. The last slide goes through some major organelles that you need to know for your first Bio 107 test. The nucleus, again, the control center of the cell where the chromosomes are located. Nucleolus, this is what's manufacturing the ribosomes for the cell. Chromosomes, the genetic material found in the nucleus. Mitochondria, ATP producer. Ribosomes, protein synthesis. They can be free or attached to the rough ER. Our RER, rough ER, modifying, sorting, and transporting proteins. Smooth ER, synthesis of fatty acids and steroids. Our Golgi complex is sorting and packaging proteins into vesicles, export from the cell or use within the cell. Here's our lysozymes, our vesicles filled with digestive enzymes. Our cytoskeleton provides movement and shape for the cell with protein filaments and tubules. Our centrosomes, these are mitotically active spindle fibers for spindle growth. Cilia and flagella, surface projections of microtubules. Cilia move fluid over cells surface, and flagella moves the entire cell. So again, just a review of what we've talked about at the beginning of this lecture.